Addiction to drugs leaves us in a dark place. First, it was just for fun. It made you feel more confident, more energetic, the life of the party. Then one day, you can't live without the drug. You get desperate for a fix, and the feeling only gets worse the longer you go without it. The drug becomes more important than anything else. Your job, your family, your friends, your life. Your life becomes something you no longer own. The drug owns you. This is the gloomy outlook for people who are addicted to cocaine. Welcome to CU Science Update. I'm Joanna Merrill. DrugRehabs.org reports that there are over 2 million cocaine addicts living in the United States today. Many who try and quit often relapse. According to CocaineAddiction.us, only 1 in 4 people quit cocaine on their own, and about 80% of those trying to quit experience a relapse in 6 months. In this edition, we will take a closer look at how occasional drug use can turn into an addiction, how cocaine affects the brain, and what research is being done right here at CU that will help us understand why cocaine is so addictive and why this research could lead to new hope for addicts. There are 2 classifications for drugs. First, ones that are legal, the kind that doctors prescribe to their patients. And then there are the illicit drugs, the ones that are considered illegal under U.S. federal law. These drugs include marijuana, ecstasy, methamphetamines, and of course cocaine, which is considered one of the most addictive drugs. Its history goes back over 1,000 years, originating in South America, where the indigenous people chewed cocoa leaves, which provided nutrients and several alkaloids, including cocaine. Even today, some South American communities still chew the leaves, claiming they give them energy and strength. German chemists first isolated the cocaine alkaloid in 1855. Almost 20 years later, cocoa leaves were shown to have analgesic or pain-relieving properties. While there were scientists who conducted research to find out if cocoa leaves had any medical benefits, others saw the leaves as having economic potential. In 1886, a pinch of cocoa leaves was included in the original recipe for Coca-Cola, although no one knows for sure just how much cocaine was put in the drink at the time. It wasn't until 1906 when the U.S. Pure Food and Drug Act forced Coca-Cola to remove the cocoa leaves from the drink. In various forms, cocaine was once used in cigarettes, as a powder, and even as a mixture that was sold complete with a needle to inject it into the user's veins with the promise that it would make the coward brave, the silent eloquent, and render the sufferer insensitive to pain. In Mississippi, cocaine was once sold in neighborhood drugstores at 5 or 10 cents for a small box. Years later, the Harrison Narcotics Tax Act outlawed the sale and distribution of cocaine in the United States. It wasn't until 1970 when cocaine was considered a controlled substance. Colombia is the world's largest producer of cocaine. They provide the world with around 600 tons of cocaine, half of which is consumed in the United States alone. On the streets, cocaine has many names, including coke, yayo, blow, or snow. Cocaine is consumed through several routes: oral, intranasal, intravenous, or inhalation. In other words, cocaine can be eaten, snorted, injected, or smoked. The reasons for trying cocaine vary. Curiosity, it feels good. Peer pressure, it's fun. Instant gratification, it blocks pain, and to simply get a rush. But as users continue to consume cocaine, addiction sets in and the urge to keep using becomes more intense. Today's researchers are finding out that prolonged use of cocaine can actually change the brain itself. For instance, brain imaging studies have shown that habitual use of cocaine changes areas of the brain that are critical to judgment, decision-making, learning, and memory. Changes that control the way the brain works. Hold up! How does the brain work? It's complex, extremely complex. Our brain weighs about 3 pounds and contains an estimated 100 billion cells. Most of these cells are called neurons, which are basically on-off switches like the ones we use to turn the lights on and off. When a neuron is switched on, it shoots an electrical impulse along a wire called an axon, the end of which shoots out a chemical. This chemical crosses a gap called the synapse, where it triggers another neuron to send a message. Different neurons spit out different chemicals that we call transmitters, which have names like serotonin, norepinephrine, or dopamine. Dopamine is a chemical that controls movement, emotional response, and the ability to experience pleasure and pain. Dopamine is also responsible for making a person feel the effects of cocaine. So how does cocaine change the way neurons send messages? Once dopamine is released, it travels through the synapse to the dopamine receptors on the receiving neuron. A protein then pumps the dopamine back to the transmitting neuron, where it gets recycled through the dopamine transporter. But cocaine blocks the dopamine transporter and prevents the brain from recycling the excess dopamine. The brain assumes there is no dopamine and starts producing more. As a result, the synapse gets flooded with extra dopamine, and this creates euphoric feelings greater than any natural high. The influx of dopamine happens in two specific areas of the brain: the ventral tegmental area, also known as the VTA, and the prefrontal cortex. So once cocaine gets into the brain, it can actually affect your ability to make decisions. When addiction sets in, the decision between use and responsibility becomes blurred. CU Science Update went to the Institute for Behavioural Science on CU's East Campus to take a closer look at the current substance abuse research being done.
On CU Boulder's East Campus is the Institute of Behavioural Genetics office and laboratory. Within these walls, research is being conducted to better understand the relation between genes, brain activity, and risk behaviors. Public access to the laboratory is normally forbidden. However, CU Science Update was allowed inside for an in-close look at how the research is conducted. Those who enter are greeted by rows of contaminant-free lab shoes and a sign explaining the animal facility rules. These guidelines are to ensure that outside stimuli do not affect the test environment for the animals. In one room, the researchers are in the process of constructing a new animal testing apparatus. In the adjacent room, the rats' cages sit empty as the rodent occupants rigorously complete cognitive tests in their study machines. The test, originally designed for primates, is used to assess the rat's short-term memory. A brief tone is played for the rat that either signals for them to press their lever immediately or after a short delay period. Rats that correctly pull their lever are awarded with a few drops of sucrose solution, better known as sugar water. The test also measures their motivation and impulsivity levels, which affect drug-taking behavior. Across the hall, other rats are testing in a near pitch-black room to better suit their nocturnal habits. These rats have been trained to self-administer cocaine by sticking their nose in a small slot. When they trigger the slot, a light shines to notify the rat of the dose. At the same time, a machine delivers an intravenous mixture of liquid cocaine into the rat's heart. The goal of the test is to get the rats addicted to cocaine in order to study its effect on the body. The addicted rats' brains are then examined using a fluorescent microscope, which can be used to determine the anatomical changes caused by the drug.
But animal research is only one part of the study being conducted in these buildings. In Research Laboratory Number 4, researchers are working on an easy and portable method for drug-level analysis that has many potential uses in the future.
So we're also developing a system here to where you can take a picture of a drug test strip and measure the amount of drug that was actually in the person's system just based off of the amount of pixels, density of pixels on the drug test strip. And that can be used in the future in workplaces and maybe in law enforcement to actually quantify or measure the amount of substance that somebody was taking at the time when they were tested for that substance.
With the Institute's simple iPhone app, the results are just a few easy steps away, making the drug quantification process much more convenient than in the past.
Work being done at CU's Behavioral Genetics program is offering insight for addicts. Dr. Don Cooper is an associate professor at the Institute of Behavioural Genetics in the Department of Psychology and Neuroscience. He is also the co-director of the Neuroscience undergraduate program at the University of Colorado. Dr. Cooper's work is focused on mapping the decision-making pathways in the brain. His research on these pathways and how they influence decision-making will hopefully shed some light on how addiction affects the ability to make responsible choices. CU Science Update correspondent Angelica Kalika met with Dr. Cooper in our studio to discuss his current research as well as the grant he received from the National Institute of Health.
Thank you for joining us, Dr.
Thank
Cooper.
you.
How did you get these grants to begin with, and really what's your main goal to use this money?
Well, it's a process that's it's kind of a lengthy process. I applied for grants at the National Institute of Health and National Institute of Drug Abuse, and attained a career award with that. And that was designed to look at the sort of molecular mechanisms of addiction. And another grant was to look at some working memory processes and how it's altered by cocaine. So using animal models.
Can you go into the animal models?
We train rats and mice to self-administer cocaine. So we hook them up with an intrajugular catheter, and they're allowed to administer cocaine. And after a few weeks, we then use various electrophysiological techniques to measure the neural activity coming out of the regions of the brain that are important for decision making, like the prefrontal cortex. These are areas that are conserved and they're utilized also in humans. So if we can understand the processes of how neurons adapt and the kind of plasticity that they show in response to cocaine, if we produce a cocaine addicted animal, and we study the physiological plasticity that results, then we're using techniques that enable us to zero in on the exact molecular mechanism. And if the idea is that if we can really understand the molecular mechanism for how neurons change their ability to code information, then that would translate into some benefit, and we would be able to use that information to address the problem in humans.
Would you help drug addicts in some way? How does this research sort of proceed from that?
Well, I think that the main thrust of the work is just at the very basic level, trying to understand how neurons code information, and that's something that we don't really understand. At the human level, we have sophisticated over the past 20 years. We've had a rise in brain imaging technologies, and that's given us a lot of insight into which brain regions are coding what. But in order to really get into the processes, you have to go a little bit more invasive, and for that we use the animal models. And for that we can actually record from individual neurons, and we can measure how they communicate with one another, a single neuron, how it communicates with another neuron. And we can ask very sophisticated and very mechanistic questions about what contribution does a single neuron, how much contribution does it play in a decision-making process. So, and that's where we're at right now, is we're trying to understand how individual neurons can store memory. And the idea is that if we can understand that, then we'll have a much better opportunity for designing either behavioral therapies or pharmacotherapies that would reverse some of the plasticity that's seen with addiction. And an example of that is cocaine addicts show that what's called a hypofrontality, which is a reduced function of their frontal cortex. And that is associated with decreased decision-making ability. So their ability to make decisions based on positive and negative outcomes is impaired. And so what we're trying to understand is how is it that this whole brain region shows reduced functionality at the cellular level. And we see the same thing in rodents. We see this reduced functionality.
Okay. So is that why you chose cocaine, because you wanted to study specifically decision-making and the neuron effect from cocaine?
Cocaine is a kind of a prototypical drug. It's got a very specific mechanism of action, so it's something that's kind of a gold standard for people to use when they want to study addiction. It's powerfully addictive in all species, and it activates the dopamine reward system. And that's something that we use cocaine because like all drugs of abuse, increase the dopamine system. They increase dopamine transmission. And so rather than studying a drug like alcohol, which has several different mechanisms, and it's not really clear exactly how it's working, we're using something that's very specific. It works in a very specific circuit. So that helps us understand the mechanism.
So it basically just narrows down what you're trying to focus on, research and study.
Yeah, within we can study a very specific circuit. When we're studying cocaine, we know all of the various brain regions that are important for that, and we also know that rats and mice, they show the same sort of patterns of intake that humans take. So over time, they tend to escalate their intake.
They become addicted.
They become addicted, right? They escalate and how much cocaine they'll take over time. They show a transient withdrawal from it, and they show high relapse potential. So if they're stressed, if you give them some kind of an acute stressor, they'll go back to they'll relapse effectively after they've been trained to no longer take cocaine. So now that if we can model the various aspects of how does an individual go from experimentation, where you first take cocaine, to the point where you've transitioned to stable intake, to the point where you're trying to quit, and then you have a period of abstinence, and then what are the factors that then cause someone to go back and start reinitiate drug taking? We can model all of that in an individual rodent, and then look at all of the genes and all of the proteins and all of the neural circuitry changes that accompany each phase of that.
That's great. That's really fascinating research, and sounds like it's going to have a lot of applications in the future with drug therapies.
Potentially. There isn't a good therapy right now.
That's true. I'd like to get into cellular memory, and what is your research with that right now?
That's something that we just recently published on, and it was, there had been a debate for a while. There were two camps on this idea of short-term memory. And when I'm talking about short-term memory, this is memory that lasts for seconds to minutes. And it's the kind of memory that we call working memory, which is, if I were to give you an analogy, it would be like if you're in the middle of a conversation and your cellular phone rings, and you take a break to answer the phone. You tell the person that you'll call them back in a few minutes. You hang up, but then you come right back to the conversation you were having, and you return to exactly what you were saying. So there had to be some sort of memory process that maintained your ability to focus on what the conversation was, despite this transient distractor. And this kind of memory is what really gives continuity to our lives. Because if we didn't have this kind of thing, we would become distracted, and then we would not be able to return back to what we were talking about, and so it would be very disruptive. And there are certain pathologies where this is the case, where, for example, in some types of schizophrenia, there is psychosis where stimuli that are around us that we normally we habituate to, we get used to, and we ignore, and that could be a siren that's going on outside, or it could be almost anything, people walking by in a hallway. These kind of stimuli we don't attend to when we're attending to something else, something that's drawing our attention, like a speaker. But for someone who has psychosis, these things are very distracting, and they orient to them. They shift their attention back and forth between all of these various stimuli, and it distracts from their ability to focus. And if all of these things are very salient to them, then it becomes very upsetting. And so this kind of process, this ability to maintain focus amidst distractors and to pick out which signals are important amidst noise, is something that's very important for proper functionality and also for proper decision making. So if you had to decide between two options, you have to be able to weigh all of the pros and cons together in some way so that you can say, well, it looks like I should make this decision. Mm-hmm. And this is a problem that drug addicts tend to have, in that the cues related to the drug taking become so powerful that they don't consider the negative consequences that could happen if they pursue that, such as getting in trouble with the law or losing their family, etc. Mm-hmm. So we think that this, at a fundamental level, there's a problem with their decision making, and it's their ability to maintain this working memory. So the debate that's happened that's been going on is, well, how does the frontal cortex, how does it accomplish this working memory?
And then how are we going to use this in the future? How do you see this research going into something, someone say, I'm going to use this to improve my life?
So, well, what we've found is that the individual neurons can store transient information, and this was something that was one of the two camps. There was a camp that said that you need a network that maintains it, and another camp that said individual neurons can do it in and of themselves, although they're participating in a network. So what can come out of this is now a focus on, we identified a couple ion channels and protein cascades. One of them was called the metabotropic glutamate receptor that's involved with maintaining an intracellular cascade that would strengthen the ability of neurons to hold memory longer.
Okay.
So for example, we have been looking with some of my colleagues at the Institute for Behavioural Genetics. We are looking at gene polymorphisms in humans that are in the genes that we found that control the cellular memory.
So
and
making memory stronger, basically.
Right, where we're finding that there are certain genetic mutations that people have, just normal mutations, that could convey a good working memory or poor working memory. And we think that if we can really understand those genetic determinants, mm-hmm. that we can then develop maybe pharmacogenetic strategies where you could boost somebody's working memory, and that would translate into improving their overall decision making.
We are hopeful that the research being conducted by Dr. Cooper and his associates will one day lead to a more in-depth understanding of cocaine addiction in the brain. Last year, researchers at the University of Wisconsin-Milwaukee made discoveries that could lead to the first ever pharmacological treatment for cocaine addiction. Propranolol, a common beta blocker used to treat hypertension and anxiety, appears to be effective in preventing the brain from retrieving memories associated with cocaine use, a major reason why users find it difficult to kick the habit. Understanding the neural mechanisms behind addiction could help boost effective treatments and develop long-lasting, if not permanent, solutions for recovering addicts. I'm Joanna Merrill. Thanks for tuning into this edition of CU Science Update.
CU Science Update is produced by University of Colorado at Boulder, Journalism and Mass Communication. To download this episode and others from CU Science Update, visit the podcast section of the iTunes Store. Or you can visit our website at cuscienceupdate.com. Finally, don't forget to like us on our Facebook page, where we post the latest in science happening in Boulder and beyond.